Welcome to the Uniformed Services University. My name is Dr. Lydia Helwig, and I am the Longitudinal Course Director for Genetics and Genomics in the School of Medicine. I am thrilled that you will be joining us this fall at USU. In preparation for starting your medical school journey with us, I will briefly be reviewing some genetics concepts and providing some helpful hints regarding navigating this content moving forward. You'll be encountering genetics and genomics throughout your curriculum here at USU, and of course in the clinic as well. The overall goal of our content is to equip you as a future provider with the knowledge, skills, and resources to use genetics to positively impact patient healthcare. Genetic information is just another important tool in our toolkit that we can use as providers to help accurately diagnose and manage our patients. Importantly, you can expect that we are going to build upon the core principles of human genetics to understand its relevance and application in clinical care so that you're able to determine when genetic testing is important to care and how to use this information to positively impact patient care and their families. There are medical providers that have specialized training in clinical genetics, including genetic counselors and medical geneticists. In addition, clinical molecular geneticists have specific training and expertise in interpreting genomic data. However, genetic testing and genetic information is something that all healthcare providers across clinical specialties will need to know how to navigate and use. Although some genetic conditions can individually be very rare, collectively genetic conditions are quite common in the general population. Due to advancements in testing technologies and in our understanding of genetics, we're able to better use this information in medical care. In fact, genetic testing and genetic counseling are standard of care recommendations in clinical guidelines across almost all specialties of medicine. This is because this information can impact diagnosis and medical management recommendations, including screening, medication choices, and other risk-reducing options. There's actually evidence showing that using genetic information appropriately can be life-saving. To begin, let's review some of the terminology that we will be using and building upon as we discuss genetics in medicine. Genotype refers to the information encoded in the genome. So for example, over here, you see a particular gene called LDLR. This is a gene that's associated with the condition of high cholesterol in families. And this is a specific genetic change that we know causes that condition of high cholesterol in families. And so this is an example of genotype information. Whereas phenotype is the expression of the genotype as a morphologic, clinical, cellular, or biochemical trait. So you can see here, this particular genetic change is actually associated with high cholesterol that you can see. This terminology is important as we consider many additional concepts in genetics, including mechanisms of disease, inheritance patterns, and interpretation and clinical applicability of genetic testing information. For preparation for medical school, you've likely already learned quite a bit about the basic principles of genetics, including how the instructions needed to direct the activities in our body are contained within our DNA sequence. You'll learn more about these biological mechanisms and what happens when these processes don't go according to how we would expect. But as a quick reminder, DNA is made up of chemical units called bases. These include adenine, thymine, guanine, and cytosine, or A, T, G, and C. In complementary DNA strands, A matches with T and C matches with G, and these form base pairs. The total composition of our genetic material in our cells, which is about 3 million base pairs, is packaged into units called chromosomes. Each of our chromosomes contains many genes, which are really the functional units of heredity. Genes are comprised of sequences of bases that encode instructions for how to make proteins. I think of our genes kind of like the instruction manual for telling our bodies how to grow, develop, and function. Really only about 1 to 2% of our entire genome encodes proteins. Currently, we know of about 20,000 protein encoding genes, in which disease-causing genetic changes can lead to over 7,000 conditions that are listed in the online Mendelian Inheritance in Man, or OMIM database. This graph really shows how much we are learning over time about gene and phenotype relationships. In humans, we have two sets of chromosomes, one inherited from our biological father and one inherited from our biological mother. Each cell typically contains 23 pairs of chromosomes, 22 of which we call autosomes and are alike in males and females, and one pair of sex chromosomes. These are XX or XY. One of each of our sex chromosomes are inherited from our biological father and our biological mother. As you can see, sons must have inherited one Y chromosome from their father and inherit one of the two X chromosomes from their mother. Similarly, biological fathers always pass on their one X chromosome to their daughters. This information will be important to consider as we discuss potential risks of inheriting a specific disease-causing genetic change and whether the specific variant is located on an autosome or a sex chromosome. Members of a pair of chromosomes typically have the same genes in the same order. At any specific locus, the two chromosomes may be identical or may have variation. The different versions of the gene are called alleles. When two different sets of alleles are present at a locus, a person is said to be heterozygous. You can see here, this can be big B and little b combinations inherited from mother or father. When a person has a pair of identical alleles at a locus encoded in nuclear DNA, they are said to be homozygous. This would be inheriting big B from both mother and father, or inheriting little b from both mother and father. Determination of heterozygosity or homozygosity is important to consider in terms of the specific trait or disease of interest. Some traits are dominant, which means that if an individual has only one copy of a disease-causing variant from one parent, that is enough to express the trait or cause the condition. In contrast, some traits are recessive, which means that two copies of the disease-causing variant, one from each parent, are required to express that trait or cause that condition. If an individual has only one copy of a disease-causing variant in the setting of an autosomal recessive condition, they are said to be what's called a carrier, and they're not expected to have signs or symptoms of the condition themselves in most cases. We will be considering genetics principles and concepts in terms of their application to clinical care. Genetic information can give us powerful information about the biological basis of a health condition that our patients may already have or may be at increased risk to have in the future. Examples of this can include diagnostic testing, in which information is used to help arrive at or inform a specific diagnosis, and predictive testing, in which an individual does not yet have a clinical condition, but may have an increased risk to develop it in the future. So again, this information can help guide medical management strategies aimed at either earlier detection or decreasing the risks of serious complications. Pharmacogenomic testing can help guide medication dosing and management for patients. Additionally, some genetic testing can specifically inform treatments, ranging from specific medication use, surgical decision-making options, as well as in some cases, even gene therapy options. Genetic testing can also be used in the reproductive setting. Some genetic changes can cause disease or other health conditions. When we look for genetic diversity, we are comparing an individual's genetic information to a reference genome sequence. This is a designated most common sequence or arrangement in a population at any one position. The reference genome is subject to constant evaluation and change. When we're looking at genetic variants, it's important to note that genetic variants can differ by the size of the altered DNA sequence. Some genetic changes can be small, Alterations of the sequence of DNA involving substitutions, deletions, or insertions range from a single nucleotide variant up to larger insertions and deletions. So an example of this, it looks like this sign right here. Normally we would expect this to say BUMP, but in this case, there's been a mix-up and there's an M where the U should be and a U where the M should be. And this changes the spelling of this word and it changes the meaning. And so this is just an example of what these types of variants might look like. Some genetic variation can also be quite large. Variation in chromosome number that leaves chromosomes intact but changes the number of chromosomes is called aneuploidy. So you can see here, this is some errors in assortment, and this can cause trisomy, where there's an extra chromosome, or monosomy, where they're missing a whole chromosome. Alterations that change only a portion of the chromosome and might involve an unbalanced change of a subchromosomal segment or a structural rearrangement involving maybe parts of one or more chromosomes are called regional variation or copy number variation. To look for these different types of genetic variations, there's many different methodologies. In order to select the most appropriate test in a given situation, it's important to understand the mechanism of disease for the specific condition of interest and the different types of disease-causing variants. I like to think about genetic testing kind of like looking at a library. We can take a close look at a specific gene, just like we can pull and read a specific book. We can also take a look at a whole section of the library to see if there are a bunch of books missing, or maybe the books are in the wrong place in that section. That's kind of like taking a broader look at an entire chromosome. Also, we can look at the entire genome, kind of like taking a look at the whole library. We will be reviewing these and other testing methodologies together further in the fall. The reason this is all important is once we're able to identify a specific genetic variant, we need to make sense of it. Not all genetic variation leads to an increased risk to develop disease, even when you're looking at a gene that may be associated with that condition. We all have lots of variation in all of our genes. The challenge is really trying to figure out which variation causes disease and which do not. The American College of Medical Genetics and Genomics and the Association for Molecular Pathology have guidelines for interpreting genetic variants and classifying those. These guidelines use multiple different types of evidence to arrive at a classification. We'll be discussing this in greater detail together. Ultimately, our goal is to apply genetic information thoughtfully to optimize care and improve outcomes for patients and their families. This includes using genetic information to guide diagnosis, inform management decisions, and support family counseling. In summary, we talked about how all healthcare providers will be interacting with or using genetic information in some way in clinical care. Throughout your curriculum here at USU and in your clinical practice, it's important to use the fundamental principles of genetics to understand risk information in families, when genetic testing is useful, what type of genetic testing could be beneficial, and how do we interpret and use these results in clinical care. So we'll be talking a lot more about genetics and genomics as we continue our educational journey together, and I am really looking forward to seeing you in the fall.